Dr. Albert Mensah is an internationally recognized physician specialist in metabolic treatment approaches for patients with developmental, behavioral learning, and mental health issues. He is the president and co-founder of Mensah Medical, a biomedical outpatient clinic of physicians and nurses who encompass the best of traditional medicine and natural medicine based on biochemical evaluation, evidence-based research, and clinical experience. Dr. Mensah utilizes a non-drug nutrient approach targeted to correct biochemical imbalances that may be associated with anxiety, fears, autism, ADHD, learning disabilities, eating disorders, bipolar disorder, depression, school phobias, mood swings, aggressive or violent behavior, childhood and adult schizophrenia, Alzheimer's disease, and Parkinson's disease. Patients can be seen at our main clinic located just outside of Chicago and in select cities at U.S. outreach clinics located near San Francisco and Los Angeles, and in Annapolis, Maryland, and Scottsdale, Arizona. Dr. Mensah.
Thank you, Dana, and good evening, and welcome to our webinar titled MTHFR and Mental Health, Understanding the Overall Effect of Individual Genetic Mutations, also known as SNPs or SNPs for short. First, we would kind of like to recognize that quite often we talk about the brain being a biochemical factory. Given the proper supply of building blocks and cofactors, processes that are absolutely essential for normal brain function. Serotonin, dopamine, norepinephrine, and other neurotransmitters are actually synthesized, or they're made in the brain. I know some of you out there are thinking, well, what about the gut? You know, there's serotonin made in the gut. However, the serotonin that is made in the brain is actually the serotonin that is utilized by the brain. It is made by a very distinct process that is separate and distinct from the serotonin made in the gut. So we have to be careful about presuming there's transfer between gut serotonin and brain serotonin. That's a topic for another discussion. I know a lot of people out there are going, ooh, I heard differently. The raw materials for neurotransmitter production are nutrients like vitamins, minerals, and amino acids. Genetic expression, or the production of key molecules that will actually move neurotransmitters from the area where they're being utilized, is enhanced by certain nutrients and actually inhibited by others. Methylation of what are called chromatin proteins, these are actually DNA proteins, is a primary mechanism for silencing or quieting genes that produce neurotransmitter transporters. By understanding the methylation cycle, by understanding SNPs as well as enzymes, advanced nutrient therapy aims to heal the brain and correct methylation imbalances. Why is methylation important? The methylation cycle is basically essential for good mental health. Basic nutrients are necessary for normal function of this cycle. Loads. It's too much of certain nutrients, and deficiencies in other nutrients may actually disrupt methylation pathways in the brain. Additionally, severe oxidative stress can lead to poor immune function as well as disruption of the methylation cycle. Let's explain. The body's methyl groups actually turn genes on or off by affecting the interaction between DNA and the cell's protein-making machinery genes. Too much or too little of important methyl groups can cause methylation imbalances. Altered DNA methylation, that is what we call epigenetics, during early fetal development can predispose individuals to not only cancer, depression, schizophrenia, autism, and other disease conditions, including Alzheimer's. So methylation is a very, very important process throughout the life cycle of the human being. DNA methylation. We all share about 99% of our DNA with everyone of the same gender. It's that 0.1% that actually makes us different. More than 10 million single nucleotide polymorphisms, we call them SNPs for short, have been identified in the human genome. These are also called gene mutations. So let's put that another way. There are more than 10 million different gene mutations that have been seen in the human genome. Most humans walking this earth today have more than 1,000 SNPs, or genetic mutations, and most any physical or cognitive difficulty despite these mutations. Ladies and gentlemen, that was a very, very important statement. I want everyone out there to understand that we all have genetic mutations. The average person has 1,000 genetic mutations, or SNPs, and most of them have no severe functional effect on either physical or mental processing. Okay? Now, there are SNPs that tend to reduce methylation, and there are others that tend to actually increase methylation. So SNPs can move in either direction. These mutations can cause our systems to move in either direction, that is, to reduce methylation or to increase methylation. It's a patient's methyl status that depends on the overall combined impact of these SNPs. Let's talk about the truth about genetic mutations, or these SNPs. First of all, I want everyone to understand the impact of a single or an individual SNP actually varies from one person to another. It is not necessarily a consistent element producing the same result from person to person. Certain strategically placed SNPs can significantly weaken enzyme function, and that is a very important point as well. However, please note, point number 3, most SNPs have little or no effect on total enzyme function. Now, all you folks out there who've been ordering all these genetic tests, I'm really talking to you here. I want you to understand one more time, most SNPs actually have little or no effect on enzyme function in general. Another point I want you to understand, and this is one of the reasons why most SNPs may have this little or no effect, the larger the molecule, the larger the enzyme, the less significance that a single SNP has on that enzyme's function. The smaller the molecule or enzyme, the greater the significance of a single SNP that may have, or may be present, having on enzyme function. So folks, one way to put it is, size does matter. Now, let's take a look at a construct here, an illustration point. This basket with the balloons flying high, or just flying, represents the MTHFR enzyme, or the methyl tetrahydrofolate reductase enzyme that many people are truly so concerned about. Now notice all the balloons. Each balloon represents a part of the total molecular weight, represents a large enzyme. Okay? Now, let's see what happens if we pop a few balloons here. The number of balloons we just lost may represent SNPs, or genetic mutations that alter the function of the enzyme. But notice, the basket is still afloat with the balloons that are present. We have had SNPs, but we don't have any total functional change in the activity of the enzyme. The basket is still aloft. Let's look further on. As we lose more balloons, or have even more SNPs, let's say this represents a homozygous SNP in the MTHFR enzyme, and please still note, the basket is still aloft, the balloons are carrying the weight of the basket, it is still a functional protein, okay, or enzyme. Next slide, please. Now then, let's look at a different enzyme. We'll just call it enzyme X. Notice this is actually a smaller enzyme. It has a very small molecular weight, and it only has 3 balloons carrying it aloft. Now, let's see what happens when we have a SNP or a genetic mutation in one of those base pairs, or one of those balloons. When we lost the balloon, we saw that the basket actually became lower. It was losing functionality. It's heading towards the ground. That was just one molecular change. Let's move to the next slide. When we lose a second balloon, we see that the enzyme activity is decreasing because the balloon is going lower and lower. It's not holding up. Next slide. In fact, the functionality of that enzyme actually becomes zero because it had a very, very small or low molecular weight, and every mutation in had a huge or significant effect. Next slide, please. Let's talk about the chemistry database and evidence-based research. We have a massive chemistry database thanks to Dr. William Walsh of the Walsh Research Institute. It contains more than 3 million chemical tests for patients diagnosed with autism, ADHD, depression, bipolar disorder, schizophrenia, and behavioral learning disorders. There are striking blood-urine chemistry differences between these patient populations and normal controls. Methylation status has been determined for over 30,000 actual mental health patients over a 30-year period. Now, let's talk about methylation disorders. They're really of two types. One of the things I really wanted to share with a tremendous number of people is that undermethylation, many people don't realize that there's also overmethylation. Not just undermethylation as being a key, there's overmethylation also to be considered. Okay? Did you know, in the general population, 70% of us walking around today have absolutely normal methylation processes going on? Now, in that normal population, we have 22% of individuals who are actually undermethylated. Forgive me, not the normal population. In the general population, 22% of people are actually undermethylated, and roughly 8% are overmethylated. Now then, when we're talking about mental health, about 70% of persons with mental health disorders exhibit a serious methylation disorder. Note, I did not say they were undermethylated. I said they had a methylation disorder, which means it could be either undermethylation or overmethylation. Let's look at the incidences of undermethylation. In the autism spectrum disorder, we note that roughly about 98% of children in the autism spectrum are undermethylated. As far as antisocial personality disorders are concerned, roughly 95% of those individuals are undermethylated. With reference to schizoaffective disorder, roughly about 90% of these individuals tend to be undermethylated. Individuals with oppositional defiant disorder, oftentimes it's about 85% where we're seeing undermethylation. With anorexia, it's at least 82%. Depression, big population of the world talks about depression. In depression, is represented by only about 38% of the population. So here's the other thought, folks. If depression is represented by 30% undermethylation, well, what's happening with the rest of depression? Food for thought. Behavioral disorders, such as behavioral disorders and ADHD, roughly have about 37% incidences in undermethylation. Undermethylation symptoms and traits. Oftentimes we see obsessive or compulsive tendencies. There are seasonal inhalant allergy type scenarios going on. There's often a history of perfectionism. Many of these individuals have a very low tolerance for pain. Oftentimes there's a prior diagnosis of obsessive compulsive disorder or oppositional defiant disorder, that is OCD or OD. Individuals engage in ritualistic behaviors, the wringing of hands, the checking of lights, all these sorts of things. These individuals tend to be very strong-willed. This is how I like things done. I want it done this way, this is my way or the highway. Oftentimes there's social isolation. And oftentimes the social isolation tends to be related to the very strong will. Not a whole lot of us really want to be at the beck and call of someone else all the time. There's often poor concentration, endurance, low levels of serotonin. Now, these are the categories, these undermethylated folks, they tend to respond fairly well to SSRIs in general. These folks tend to be very competitive in sports, oftentimes have frequent headaches. There's often a strong family history of high accomplishment. These are the generational academics, all the PhDs, MDs in the world that span 4 or 5 generations, as far as people know. They tend to have a calm demeanor, but oftentimes very high inner tension. Many times there can be delusions or a thought disorder. Physically speaking, they may be a lot more slender, and we'll get into that, the reason for that a little bit later in this discussion. There are often phobias, addictiveness, high libido, suicidal tendencies, and non-compliance with therapies, unfortunately. Primary causes of undermethylation. Undermethylation usually results from SNPs that weaken MTHFR or other enzymes in the methylation cycle in utero. Please pay attention to this. In utero before birth. We're not talking about effects after birth. They're actually very different situations and circumstances level prior to birth in the development of the fetus versus after birth. And the MTHFR is hugely significant prior to birth, but we see things a little bit different after birth. Enzyme mutations, or SNPs, in the methylation cycle are also very, very important. Not just MTHFR, but the methylene synthetase enzyme or the COMT enzyme, just to name a few. We can also see histamine overload. Histamine and methyl have the exact opposite relationship. So when you have high levels of histamine, it actually sequesters or pushes down concentrations of methyl. So histamine overload can be a primary cause of undermethylation. We also know that protein deficiencies or malabsorption can also contribute to undermethylation. Here we have the methylation cycle, and just to sort of point out some of the key enzymes that individuals are concerned about in a lot of our testing, not just the MTHFR but also the 23andMe. We have the MTHFR enzyme here, the methylene synthase enzyme here, and a slew of other enzymes that, roughly speaking, are all involved in these pathways, and quite a few enzymes that actually aren't listed here. But what I want to point out is, here, when we look at what happens between SAMe and S-adenosyl homocysteine, what we see is the production of methyl. This is the key thing that so many people are fussing over. The difficulty is, we want to emphasize it's the absolute concentration of methyl that determines whether you are undermethylated or overmethylated. It's the sum total of activity of all of these enzymes that determine your methyl concentration status and therefore whether you're under normal or overmethylated. It is not the activity of one singular enzyme. Incidences of overmethylation. In panic or anxiety disorders, now pay attention to this one, 64% of individuals with panic or anxiety disorders are actually overmethylated, not undermethylated. Paranoid schizophrenia, not all categories of schizophrenia, but in the paranoid schizophrenia types, 52%, that's a little bit more than half of these individuals are in the overmethylated category. ADHD, 28% of individuals with ADHD are actually overmethylated, not undermethylated. Disorders, roughly about 30% of those individuals are actually overmethylated. Now depression, a very interesting one, because we've based so much of our work, are discussions on the idea that undermethylation is the key to depression in almost everyone. Well, no. In 18% of individuals, at least 18% of people with depression are actually overmethylators. Now, what's important about this piece here is that these are the individuals that have a very negative response to SSRI medications. These are the individuals that are susceptible to increased tendencies of suicidality or even homicidal behavior a few weeks after starting an SSRI medication. So it's important to truly have an accurate perspective on an individual's methylation status before starting any type of pharmaceutical in nature. Overmethylation symptoms and traits. Now, these individuals can also have high anxiety or panic tendency. Oftentimes, nervous legs and pacing are seen. Food and chemical sensitivities can be there. Sleep disorders, depression, as we've mentioned. Self-mutilation. I want to pause there for a minute. We've seen the people who cut themselves. We've seen people who, now, no one out there, don't send me letters or mail or anything because I mentioned this, but people who have multiple piercings. While we're not calling that mutilation per se, let's just say that later on we see they have a high pain threshold and they require greater stimulation than undermethylators. So high piercings, lots of tattoos, oftentimes these individuals are actually overmethylators. Mouth, and here we go. They have an adverse reaction to SSRIs. It could be something as benign as simply no effect or response to the SSRI, or it could be something as dangerous as suicide or homicidal activity because of the interaction between the chemistries of the medications and that individual's unique chemistry. They tend to be excellent in terms of socialization. They have a high empathy for other individuals. Folks, I've got to tell you, if you're ever in the hospital, you would like your nurse and your doctor to be overmethylators. Well, maybe your nurse anyway. You want your doctor to have probably normal methylation status. But those who are going to give you care are usually very, very powerful in overmethylation. We do tend to see a tendency of postpartum depression with overmethylators as well. But there are other factors such as copper toxicities that can contribute or might be the more significant key to postpartum depression. Oftentimes, there's a very high pain threshold, as we mentioned before. These folks tend to be low in motivation as far as school is concerned. Now, let me clarify that a bit. It's not that they're not intelligent. As a matter of fact, many of these individuals who are overmethylated are geniuses. But they're geniuses that most of you out there have heard as being, well, they score well on tests, but they're getting C's and D's in school. Because they're highly intelligent, they get it, but they're not quite so motivated to do the work all the time. So their grades don't truly reflect their cognitive capacities. These individuals, unlike the undermethylated, tend to have an absence of seasonal allergies. They can be very artistic or high in musical capacity or musical ability. These are the individuals who really color the world for us. They're the artists, the musicians, the philosophers, the poets. Many times we call them the people who march to a beat of a different drum. Now, on the negative side, we might see some paranoia. Hyperactivity is also seen in overmethylated individuals. Unfortunately, a lot of times they can fall into the category that, well, they believe that everyone thinks ill of them. They tend to have obsessions, but without the compulsions. Oftentimes they have low libido. And here's the deal. They have antihistamine intolerance. They also have an estrogen intolerance as well. Primary causes of overmethylation. Overmethylation is generally caused by enzyme weaknesses, or SNPs, in the SAMe utilization pathways. That is, what's happening after SAMe is produced on its way to methyl and creatine. Impaired creatine synthesis, just kind of touched upon that for a minute, is also a primary cause of overmethylation. AGAT enzyme, or the GAMT enzyme SNPs, or with arginine or glycine deficiencies. Now, please pause. Those of you who think you're overmethylated out there, don't go out and try buying arginine and glycine and trying to treat yourself. It doesn't work that way, folks. The next point I'd like to make is that oftentimes there is impaired cystathione production or synthesis. That is a SNP in the CBS pathway. Also, methyltransferase SNPs, in other words, more of the enzymes that actually transfer methyl groups from point to point, can have mutations that lead to overmethylation. So when we talk about utilization of SAMe, SAMe that comes from the methylation cycle, most of it is actually going to creatine synthesis. 70% of SAMe from methylation goes into creatine synthesis. Creatine synthesis then, of course, moves on to muscle production and muscle development. 30% of that SAMe goes on to other reactions. Let's talk about creatine synthesis. We see here in the left-hand column that arginine and glycine and an enzyme that is just far too long a word to speak, I'll just call it the AGAT enzyme, produce guanidino acetate and ornithine. Now, that combination, along with this GAMT enzyme, thanks to SAMe, will produce creatine, okay? And it will leave the SAH present after that. This is extremely significant because what we're looking at here is where is all that SAMe, where is that methyl going? It's going predominantly into creatine production. Folks, if you remember nothing else from this program, this is what I want you to recognize about enzyme mutations and methylation. Enzyme mutations and methylation, there is a tug of war between the enzymes in methylation pathways. This is a methylation tug of war phenomenon. We've got enzymes on the left-hand side that are involved in undermethylation. Enzymes such as the extremely large MTHFR enzyme, the SAH enzyme, the BHMT, the MS, and the MAT enzymes. All of these are key players in undermethylation. What we cover is that we focused on this enzyme and perhaps one or two of the others, and we haven't tested for overmethylation. So we assume because there's a SNP here that overall functionality is challenged and we end up being undermethylated. No. Here we see that there are enzymes involved in overmethylation as well. The AGAT enzyme, the GAMT enzyme, the CBS enzyme, and the MT enzyme are also in play. So the enzymes on the left are battling the enzymes on the right, and may the winner take over methylation. Not overmethylation, but win the battle of methylation. Now, if all enzymes are functioning properly, if all variables in the cycle of methylation is working correctly, or are working correctly, we should have normal methylation. And that's what we talked about for the 70% of individuals right now, normal methylation. But for those of us who've got challenges, whether they are emotional, cognitive, or physical, we need to understand there is an absolute tug of war, a balance between undermethylation and overmethylation enzymes and different factors or variables. Now let's talk about testing for methylation status. We tend to recommend the whole blood histamine or the SAMe-SAH ratio test as the most practical and best test for histamine. Overall methylation status is a critical component when it comes to determining one's biochemistry. The whole blood histamine test can measure the net effect of the various SNPs or mutations, whereas genetic testing focuses on individual SNPs, not the overall product of those SNPs. It just talks about which enzymes might have a problem. For example, Most persons with the 677T MTHFR SNP are undermethylated. But guess what? Many others with that same SNP are actually overmethylated. So you see, it's not about the one enzyme. It's about the entire group of enzymes all working either together or in opposition. Elevated blood histamine indicates that undermethylation, and low histamine is present evidence of overmethylation. That's because histamine and methylation have a diametrically opposed relationship. High histamine suppresses methyl, high levels of methyl suppress histamine. So histamine treatments can artificially lower blood histamine and should be avoided for several days prior to testing for your histamine status. Why not genetic testing? Well, it's not entirely a bad thing. We're not totally saying no to genetic testing, but we have to have the proper perspective about what genetic testing is actually doing for us and not doing for us relative to revealing information. Genetic testing can identify predispositions for many disorders, such as breast cancer and Alzheimer's disease. Very, very important. However, the reliability of genetic testing, such as the 23andMe test, for assessing methylation is not quite limited at present. It is not targeted to any one condition in particular. Therefore, it's not really a good guide to treatment therapies. It tells you that there are possible challenges to certain enzymes, but it's not a real guide for treatment. Now, some people out there may disagree with that. And I just say to you, well, if it were such a great guide to treatment, why aren't people doing better when being treated based upon these particular tests? And why is there so much confusion around them? Okay? Identifying SNP weaknesses in the MTHFR and other methylation cycle enzymes does not necessarily mean that an individual is undermethylated, since there are SNPs that also produce overmethylation. Okay? Folks, genetic testing is qualitative in nature. It is not quantitative. I'm going to say that again. Genetic testing is qualitative. It tells you that maybe or maybe not there's a defect, large or small, we don't really know, possible in the activity of a given enzyme. It doesn't tell you how much work that enzyme is or is not able to do. It just suggests that there may be a problem. These tests do not actually tell you how much methyl, quantity of methyl, is present at the end of the testing process. So let's just say, since genetic testing is qualitative and not quantitative, it is improbable by genetic testing alone to either determine the net methylation potential, that is, somebody who's either undermethylated or overmethylated, or to direct treatment management. Let's talk about epigenetics and methylation. What is epigenetics? Epigenetics involves the alteration in gene expression due to chemical factors in the womb and influence of environmental factors throughout life. So, sort of that discussion around nature versus nurture, socialization versus birth. It's about both, really. What our genes were designed to do in the womb, interacting with what our environmental circumstances, our stressors, our oxidative stressors, are doing after birth together. Our environment can actually affect genetic expression. We can change proteins that lead to, ultimately, to anxiety, depression, or even different disease states by way of interactions between our genes and the environment. This is what we loosely would call epigenetics. Every cell in our body has the potential for expressing any one of 20,000 plus genes in our DNA. The production of gene proteins or gene expression can be turned on or off. We call that gene silencing. These epigenetic processes are more vulnerable to environmental factors such as radiation, temperature, pesticide exposure, dietary choices, toxic metals, viruses, and stressful life events. There are many other variables as well. We can even talk about EMFs, that is electromagnetic frequencies, and sleep disturbances. Talking to several people, they'll tell you, we live in a world where the average person may have at least one television or one electronic device in the bedroom, say a TV that now has a DVR, a radio, a clock, different things that tend to emit frequencies of very light electromagnetic waves, or maybe even just the light sensors themselves can be disturbing to sleep. So there are a lot of variables to consider in our lives in terms of how we want to move through life, what is seemingly working for us and what is not. Other nutrient imbalances. A genetic or epigenetic imbalance in a nutrient can alter brain levels of key neurotransmitters and result in abnormal brain chemistry. Vitamin D deficiency has been associated with depression, schizophrenia, ADHD, and other mental disorders. And I would rather say other cognitive impairments. Mental disorders tends to have a very negative term, and it's not about some mystical thing in the mind. It's actually about a biochemical construct in the brain. Pyrrole disorder is an abnormality in biochemistry resulting in the overproduction of pyrrole molecules. Symptoms of pyroluria include severe inattention, extreme mood swings that tend to be rapidly cycling, very, very important, severe depression, high irritability, and temper. Copper overload tends to lower dopamine levels and increase norepinephrine in the brain. Imbalances in these important neurotransmitters have been associated with paranoid schizophrenia, ADHD, bipolar disorder, postpartum depression, and violent behavior. Fatty acid imbalances have been associated with depression, ADHD, schizophrenia, bipolar disorder, and dementia. As a quick reference, many of you out there have already known, heard about, and just talked about, or many of you are even using different fats now as a result of all the books that have been written about fatty acids and their significance. So we know they're very, very important. But the right fatty acids are key for the given individual. In many cases, individuals exhibit more than just one biochemical imbalance. So please don't think that any one disorder is going to fix every aspect of your life. It is not. It all depends. First, you have to find out what is out of balance. Are there nutrient overloads or are they nutrient deficiencies? Then we have to talk about metal regulation or metal dysregulation. Things like copper, zinc, manganese, all sorts of lovely metal elements that are present in the system. Gastrointestinal issues, including food allergies, sensitivities, malabsorption. All these things come into play. Of course, as well as testing for methylation disorders. And we like to use the whole blood histamine as a definite evaluation here. Pyrrole disorder is also included, looking at zinc and vitamin B6 imbalances. What's key about pyrrole disorder, I just want to say here, is that it is truly a reflective marker of oxidative stress as well as inflammation. So it's not just about being a biomarker for mood swings or mood states. It actually will reflect oxidative stress. Overview of treatment. First, you need to start with a physical examination, an extensive patient history, specialized laboratory testing with blood and urine. Diet and gastrointestinal issues also need to be assessed and addressed. Advanced nutrient therapy protocols are prescribed at the appropriate therapeutic level to target the patient's specific needs in order to correct underlying biochemical imbalances. And of course, as always, there's nurse and physician follow-up care. Well, now available, we have Dr. William Walsh's book, Nutrient Power. For those of you who have not read this book, this is a great example. It is a great key guide to how powerful nutrients are. You know, in the world of medicine, we're trained as physicians to actually believe that nutrients have a micro or minimal effect on cognitive impairments, mental health disorders, or anything else in the human body. And it's actually quite the opposite. Thanks to Dr. Carl Pfeiffer originally and Dr. William Walsh, what we're seeing and understanding is that nutrients have tremendous power. Pick up this book. It's also available on Amazon. Now, I have this slide here quite reasonably because I think many of us were actually shocked by the recent death of this lovely man that we know, Robin Williams. As an actor, as a performer, he has personally touched the lives of many, including yours truly here tonight. I grew up watching this man. He made me laugh more often than I care to really speak of. So I wanted to take this opportunity tonight to certainly say to his family, we grieve with you, and of something that we have on this screen. What we know is that depression is a flaw in chemistry, not character. So for those of you who are out there who are suffering from depression or any other circumstance or illness, please remember it's not about your chemistry, it's not about your character, it's about abnormalities in chemistry. Seek the appropriate help wherever you go. Keep working. Don't let anyone tell you that there is no solution, no improvement can occur. It can be had. And on that note, I'll say thank you very much for tuning in. And to all those Robin Williams fans, go back to Mork and Mindy, and we say nanu nanu.
Thank you, Dr. Mensah. At this time, we're now going to begin the Q&A portion of the webinar. So please type your questions into the question pane located in your control panel. Our main clinic is located 25 miles west of Chicago, and patients can be seen at our U.S. outreach clinics located in Annapolis, Maryland, Burlingame, California, and Scottsdale, Arizona. And our first question for you, Dr. Mensah, is are there any known SNPs that are associated with pyrrole disorder?
That's actually a very good question. And no, I don't know of any SNPs that are associated with pyrrole disorder at this particular point in time. But that's a very, very good question. I think one that's very worth evaluating. We've done a great job in the scientific community of identifying tons and tons of mutations, and that investigative process is still ongoing. But no, there is no revealed test to determine at which locus on the gene that pyrrole disorder might be occurring. Now, the other piece to understand is that pyrrole disorder is not simply a genetic disorder. There are two forms of pyrrole disorder. One is indeed genetic, the other one is actually acquired. Anyone can become pyroluria by physiological mechanisms of stress. So it is not simply one that anyone who has pyrrole disorder has a genetic deficiency. There can be an environmental cause with regard to pyrrole disorder. Okay?
Dr. Walsh advises that persons that have low histamine should avoid folic acid in any form. Too high a risk for methyl pocket. However, recent MGH by Rothman shows that B12 and methylfolate supplementation of schizophrenics on antipsychotics saw resolution of positive antisocial symptoms in 16 weeks. Any ideas?
Well, I'm going to ask you to repeat that question in pieces. I think that the first part was misunderstood. In individuals who are overmethylated, actually the treatment of choice is indeed folic acid and folinic acid, any form of folate is actually recommended in individuals who are overmethylated. So I think there might have been some misunderstanding with regard to that part. Now, what was the second part, Dana?
So this new study shows that B12 and methylfolate supplementation for schizophrenics on antipsychotics saw resolution of positive antisocial symptoms within a 6-week period. Any ideas?
Yes. Now, in that aspect, the schizophrenics in question, now remember, when we talk about schizophrenia, once again, that is a broad category. These individuals were probably more than likely not broken down by way of their methylation status as either under or overmethylators. The odds are they were overmethylators who were exposed to the B12 and the, what was the other?
Methylfolate supplementation.
And the methylfolate supplementation. It's kind of like when Abraham Hoffer talked about the treatment of schizophrenics and referenced niacinamide as being a great element used to treat schizophrenia. And many individuals started using niacinamide, many got great results, but a lot of others didn't. Niacinamide is also part of a protocol to treat the overmethylated individuals. So my firm belief is that the individuals who responded positively in that subcategory of schizophrenics were actually the overmethylated schizophrenics and not the undermethylated ones, or the pyroluric ones for that matter.
Since we often think of addiction as an undermethylated trait, I'm curious if you could expand upon how this is different when an overmethylated individual turns to addiction for comfort.
That is an excellent question, and whoever sent it, I'm really glad you brought that up. I want to share with you for a minute, let's talk about undermethylation. Now, the undermethylators who tend to become addicted to anything, alcohol, drugs, caffeine, whatever have you, tend to do so because there is when we talk about perfectionism in undermethylators, the undercurrent there is that there's almost this addictiveness to maximize all of your capacities. So biochemically, individuals tend to get addicted very easily because they have addictive personality. Very different than what we see with overmethylators who turn to substances. Now I'm going to talk about something that we didn't talk about in this slide presentation. Overmethylators, as far as neurotransmitter activity is concerned, tend to be high in serotonin and in norepinephrine. So what you see is that they have very high levels of neurotransmitter activity. Imagine your brain constantly being in the on cycle, never able to stop. It's going every which way but loose, and you're trying to achieve some level of calm. It's the use of these outside agents, certain drugs. What seems to be drug addictiveness is not. It's actually an attempt at self-soothing by interrupting neural pathways with foreign substances. Phenomenon for overmethylators. For overmethylators, it's actually one of trying to find peace and stopping the racing brain, as opposed to with undermethylators where it's more about addictive tendencies. Okay?
Can someone switch between under and overmethylation? Are you predominantly one or the other? I seem to have characteristics of both.
That's a great question. The answer is no. And you may have characteristics of both because in some circumstances, first of all, for example, with hyperactivity, some people may suggest that they're hyper, and in some cases you might see that with overmethylators, you might see that with undermethylators. First of all, not all symptoms are mutually exclusive to undermethylation or overmethylation. But the key thing here is that you are born your methylation status. You cannot switch. You do not start out as an undermethylator, then convert to an overmethylator. Remember, methylation, as we talked about very early, the MTHFR and other enzymes involved and SNPs occurring primarily before birth are key to determining what your methylation status is. So you're born that way. Now, what we can do is that we can use various treatment modalities to deal with the sequela of your methylation status. So if you are an undermethylator at birth, we can find ways to add more methyl to your situation so that now the different enzymes, proteins, and neurotransmitters that require methyl or methylation can all function properly. But no matter what we do, you're still born an undermethylator. Now, the other side, let's say you're born an overmethylator. Okay. Our goal then is to remove as much methyl as possible in order to help normalize your methylation. But you don't switch genetically from one to the other. Now, the real truth of the matter is, for the person who sent in that question, you could be normally methylated. We talk about the academic brilliance of an undermethylator statistically. We talk about the musical and creative capacities of an overmethylator statistically. Well, there are tons of individuals out there who are both creative and academically gifted. And when you check the methylation status, they're normally methylated. So one of the ones I really like to reference, and if this is any of you out there, please don't get mad at me, but we've got the attorneys, we've got the physicians who are there by day and by night. They are musicians in the clubs, they are playing musical instruments and having a grand time, or on the weekend, they're riding Harley-Davidson motorcycles and wearing bandanas and sunglasses. Okay? So you can have creative academic giftedness in the normal population together. It doesn't mean you're one or the other. You simply might be normally methylated.
This question kind of goes along with this. Some of the symptoms on the two lists for over and undermethylation can appear in the same individual, such as seasonal inherent allergies and chemical sensitivities. Are these symptoms lists general indicators?
Yes, they're not to be taken as 100% diagnostic. They're not mutually exclusive. And for different reasons, people may have sensitivities in one area that it's not actually related to methylation. For example, a sensitivity in the gut to particular foodstuffs may actually show up as an allergic reaction on the skin. A good dermatologist will often say that a disease or disorder of the skin is actually a manifestation of something that's going on internally and deeper. Okay. However, an undermethylator on the other side may be the one whose histamine level flares up and overreacts with any potential immunological insult or exposure. So they're not necessarily mutually exclusive, but they may be there for different reasons. The one may have nothing to do with methylation at all, but may have something to do with the immunological pathway, and the other might have to do with methylation. So there are a variety of reasons for things like sensitivities and allergies and so forth, and that's why it is that it's not necessarily 100% written in stone for either side.
How much does the father's nutrition impact fetal development and epigenetics?
That's actually a very good question. Paternal nutrition, as much as it relates to the status of developing sperm and certain immunological functions, very, very complicated issue, and one that is not purely dad's fault. We know now that the father contributes certain elements in the process of mating, and also the mother contributes some very interesting elements. For example, a mother's chemistry may actually inhibit sperm from either penetrating the egg to produce the ovum, or it may actually be extremely volatile an environment for the sperm. It's a very interesting thing when we talk about chemistry and synchronization between two individuals that meet. Two very attractive individuals can come together and find no real interest in the other person as far as mating is concerned. And yet very divergent people might come together and something may just seem right. Is that really a surface manifestation of internal chemistry making its wishes known? It's quite possible. It's not just the dad side, it's not just the paternal side, it is also maternal in nature. But to more address the question, nutrition can at any time affect a man's capacity not just to think but to function or to develop, and to develop any particular protein, enzyme, or chemical pathway or functionality anywhere in the system, including that involving reproduction. Taking care of yourself is the best way to really ensure or to maximize your reproductive capacities for both males and females.
Going along with this, then another question: how long before conception do the parents need to begin to address their nutrient needs?
That's a great question. We often actually do some not only premarital counseling but biochemical evaluations for individuals who, and we assess both males and females. And a lot of times we usually will start 1.5 to 2 years prior to these assessments, prior to individuals coming together to start to generate children. It's very important, if you're even thinking about the mating process in order to produce offspring. Now, I hate to sound clinical, but we know now not everybody engages in the whole process of marriage and so forth and so forth. But if you're thinking about children, now's the time to really start doing everything you can to maximize your nutrient capacities and anything else that might be going on. Okay? So start right away as soon as you think about it. The longer or the earlier you start, the better prepared you're going to be.
Mm-hmm. Why are undermethylators more prone to produce?
That's a great question. And here's where the construct of genetics as well as epigenetics comes into play. We know that, once again, more than 95% of children who are in the autism spectrum are undermethylated. The question is, well, where did they get it? Remember, this is a genetic or an epigenetic phenomenon, which means it's a family lineage that is putting forth this methylation challenge that gets passed on to the developing embryo. And in that sense, if one is an undermethylator, the expression of undermethylation may change. It might be that the child comes out anxious or depressed or an alcoholic or a cigarette smoker, even with undermethylation. But one of the potential challenges for the expression of undermethylation that is passed from parent to child is indeed autism. And that's one of the things we note. Now, it's not just autism. The question is really susceptibility to oxidative stress. So if the undermethylator is put together with the right sequence of oxidative stress mechanisms or stressful insults, whether it's in utero or after birth, what we see then is that incapacity to tolerate the oxidative stress is one of the major contributors to autism. So we don't want to say it's, okay, you're an undermethylator, you're going to have an autistic child. No, that's not what we're saying. What we're saying is that undermethylation that is of lineage variability passed on to the developing fetus or embryo, child at risk for a variety of different disorders, including autism. It is indeed epigenetic, environment mixing with genetic tendencies. And yes, indeed, it tends to be more with undermethylators. Now, what are we seeing, though? It's not just dad. It may not just be mom. The issue is when we often see two undermethylators coming together. And now they both are contributing undermethylated situations as far as genes are concerned. Now we may have a weakened capacity if the right undermethylated constructs are present at the union of the sperm and the ovum. Now what we're seeing is huge susceptibility to environmental stressors. So it's not just one side. Both sides contributing may be one of the truly strong predisposing factors for autism as a blueprint possibility, but with environmental insults. Now, what are those insults? Could it be extremes of folic acid being given from preconception all the way through to birth and even during the breastfeeding phase? That's a possibility. But we know that folates and undermethylation don't go together. So there is indeed that particular potential. So it's not just the genetic construct. An undermethylator can produce a child who's undermethylated, and that child could be perfectly fine. It's about environment. It's about both sets of parents. It's about genes. It's about a big cosmic soup of environmental circumstances mixing with genetic predispositions that lead to autism. Very, very complicated issue. And that summary by no means is exhaustive. Okay.
I've been asked to put up a couple of slides for those individuals that wanted to take some notes for testing. Next question is, if I have high homocysteine, does that mean that I am undermethylated?
Not necessarily, but it's a great question. A lot of the times that we've had to pause when treating undermethylation, it has occurred because individuals had high homocysteine. Now, high homocysteine really is more about, from our perspective, cardiac status. So individuals who are over 30, we pay more attention to the homocysteine levels relative to their cardiac issues than we do for anything else in terms of cognitive issues. So having high homocysteine does not mean that you're undermethylated. We see tons of people who are undermethylated with perfectly normal homocysteine levels. So the answer is no, not necessarily. But there's some individuals who are undermethylated and have higher homocysteine. But no, it is not necessarily necessary. In fact, we know statistically that it's simply not the case that having high homocysteine means you're undermethylated.
I'm homozygous 677 and a competitive overachiever with OCD, anorexia, but SSRIs have made me suicidal in the past. I also have CBS, COMT, BHMT, etc., SNPs, pretty much all of the significant ones. So what would be a good next step for dealing with my issues when there are so many factors at play?
Histamine status. See, once again, you've done all the testing that's out there for genes, and you've listed some very interesting attributes that would point you to being undermethylated or overmethylated, as a matter of fact. But tests that you've done actually give you the quantity or the concentration of methyl to really determine what your methylation status is. Remember, after you do all these genetic tests, after you test for the activity of undermethylation enzymes and overmethylation enzymes, the bottom line is, what is the sum total of methyl you've got there? If you've got a lot of methyl, after all that, guess what? You're overmethylated. If you have a little bit of methyl, you're undermethylated. The best example for quantifying your methyl is really the SAMe-SAH ratio. The problem there is that that particular test has to be done and at the lab within an hour. So it becomes not really practical for most individuals. But the histamine test is a great reflective marker for the very simply, to the person who asked the question, I would say get a whole blood histamine test, contact Direct Healthcare Access 2. They're the ones who are going to send this out to you in terms of instructions. That's the best way, get you to the right lab, and have the best results for really processing the histamine. So I would say your next step should be get a histamine test, and let's find out for sure what your concentration tendency for methyl really is. That should tell us if you're over or under methylated. And then treatment can begin after that.
Why do undermethylators tend to be thin?
Ah, I was waiting for that one. I'm glad somebody brought that up. If we let's go to the slide on the synthesis of creatine. We can go back to that one. Okay. Now, folks, if we look at the right-hand side here, creatine is that element that will later on lead to muscle production. Okay, it goes to producing thick muscles. Now, if you don't believe me, talk to the bodybuilders out there. Read bodybuilding magazines. You will see more products that talk about arginine and glycine being added into your nutrient supply in order to give you bigger muscles. What they're doing is they're taking arginine and glycine, assuming this enzyme and this enzyme are intact, and they're producing lots of creatine artificially in order to enhance muscle development and growth. Okay? Now, this part assumes that our methylation pathways with regard to overproduction. Okay? With undermethylators, remember, the blockage occurs quite literally before this aspect of the cycle is developed, and so you get a decrease in the production of creatine. So with less creatine, you don't make as much muscle. So that's ultimately why undermethylators, those individuals with very little, I wish I could draw on this, carbon, hydrogen, number 3 associated, CH3, very little methyl. With very little methyl, you cannot make creatine. Little creatine, you can't make muscle. That's why stereotypically undermethylators tend to be slight of build, versus overmethylators tend to be our big, giant, stocky, oftentimes no one sue me here, our Iowa State, Nebraska Cornhusker giant football players, okay? These mammoth walking trees with tons and tons of muscle. Those are individuals with a great amount of methyl that gets moved into creatine, which moves into muscle production. Okay, so that's why a true undermethylator, low production of methyl, tends to be slight in physique.
Histamine production is determined by the DAO/MAO and HNMT enzymes. So why does high histamine necessarily mean that you are undermethylated? Can it just mean that you have mutations in these genes and therefore can't break histamine down?
No. Histamine breakdown is very different than histamine production. When we're talking about histamine has several functions. One of them is that it's an enzyme in the GI tract. It is also an allergic reactant participant, and it is also a neurotransmitter in the brain. There's a difference in histamine's function in the GI tract from the brain. So now remember, there's several genes that are affecting histamine, not just one, of course. But the key here is the relationship between methyl and histamine. Okay? There's a relationship between methyl. Methyl that is high is going to suppress histamine concentrations. If you have low levels of methyl, those histamine concentrations can rise even more and further suppress methyl. So it's about relationship of functionality. It's not in terms of creation. It's actually more reflective about the relationship they have. Okay, it's one about one's high, the other's low, to be very simple, so to speak.
And someone also wanted to ask, what about undermethylators that are not thin?
Well, remember, we talk about statistical evidence. We're not talking about 100% absolutisms. Very rarely do we see anything that's absolute. I was talking to one lady who was inquiring about some things earlier today, and like other people have said before, the only thing that we know of that's absolute in biochemistry and life is that if you're born, you're going to die. That is absolute. If you're not born, you're not going to die. That's absolute too. However, there are very few constructs, very few relationships, very few molecular pieces that are 100% absolute. While we talk about the use of folic acid for example, in autism, we see that roughly about 25 to 30% of autistics actually respond to folinic acid very well when they're actually undermethylators. That means that there's another element in the system that's in effect that we weren't able to detect, and these individuals actually do pretty well. Okay? So nothing is 100% absolute, but statistically speaking, we're looking at the bulk of individuals who tend to fall into these categories. Okay?
I'm concerned about the long-term effect of antihistamines and overall methylation. Can you discuss this some more? Do you find more imbalances with the H3 and H4 receptors?
Good question. First of all, I want to histamines that we have on the market, and I really want to clarify the over-the-counter antihistamines versus the antihistaminic effect of SSRIs and certain pharmaceutical agents. I would be more concerned about the overall long-term effects of certain pharmaceutical agents like the SSRIs or the SNURPs, the atypical antipsychotics. These are the medications that you need to worry about. Overall, for individuals who have allergies, things like Benadryl, other over-the-counter antihistaminics actually are more functional in almost a staccato as opposed to more of a staccato utility. You have an allergic event, you take some Benadryl, you move on with life. Maybe the next day, the next week, you might have to use it. Those things tend not to be as detrimental. In general, now I'm sure we all know somebody who went literally batty after taking Benadryl and had a severe response. And yes, for some people that can be quite severe, but what I'm really referencing are the individuals who are, as a group, more so going to be susceptible. And the over-the-counter stuff typically doesn't have a major effect on overmethylators quite so severely. It's the pharmaceutical elements that you have to worry about. Those are the ones that, once again, statistically, can plunge an overmethylator into severe, severe depression and or suicidality or homicidality. Okay? That's what I'd like you to keep in mind.
It's interesting you just said that, because this person says, what if you're taking antihistamines and can't stop taking them without horrible side effects? I have tried to stop taking them, but after 1 to 2 days, not only do I have awful allergic reactions, but panic attacks. How many days do you need to stop taking them before you can test?
Well, that's a very good question. One of the things that we would say to you in terms of testing is write down everything that you're taking in terms of an antihistamine, take the test, and then what the provider does is that the provider will then extrapolate the results. We know that there are certain antihistaminics that have a greater antihistaminic effect than others. Okay. And if you tell us the duration of time you've been taking these meds, as well as which meds you've been taking, then we can sort of extrapolate to say, well, this person is probably normally methylated, or they are likely high undermethylators, but maybe not severely undermethylated, based upon what we're seeing with regard to the results and the medications they've been taking. So if you have a severe response, listen, the most important, okay, alive and well, don't get off your antihistamines if they are preserving your sanity, reactivity, or just physiological functionality. Stay on them. Take the test, and then let's see what things look like. Okay? There are ways to move around the antihistaminic medications that are necessary for you and testing. So don't be discouraged by that.
Is a single whole blood histamine test truly clinically relevant as levels naturally elevate in the morning? Or should we be doing multiple draws over a 12-hour period to determine average levels?
Good question. And the answer is no, not necessarily. As a matter of fact, most of the time you don't do multiple draws within a 24-hour period. What you want to do is that you want to take a look at the histamine level at some point in time, get one result. So let's say we draw a histamine level today. 6 months from now, if you've gotten a definitive undermethylated, overmethylated, or normal methylated answer during your first draw, you will look to treat based upon your findings, and then 6 months later, you want to retest the histamine. If you're truly undermethylated or overmethylated, what you will see are still the trends in those directions, but hopefully improvement, so to speak, in symptomatologies. 6 months later, there should be a third histamine blood draw done. That way you now have a comparison over the course of a year of 3 different histamine draws. That typically weeds out any false highs or false lows in the patterning. And then retesting on a yearly basis usually gives most people a very, very concrete answer to their methylation status in addition to the symptoms of improvement based upon the treatment that was provided, that was done. Okay, so multiple blood draws are not necessary during the course of a 24-hour period.
Lyme psychiatrist Robert Bransfeld says that those with autoimmune problems react badly to SSRIs. What do you think about pathogens, autoimmune, and psychotropics?
That's a very good question. If you're asking how does that interact with methylation, that's a very, very broad question.
She also asked another question, I'm sorry. Do you think possible pathogens, such as Lyme disease, how do they play a role in affecting methylation?
Actually, those diseases, those in environmental elements that literally trigger what is internally already in place. If, for example, somebody has a weakened immune system or a heightened reaction, basically those pathogens are stimulants to a process that is revealing what was already there as a baseline epigenetic construct. So let's say that you are actually born an individual whose methylation processes developed and blueprinted at birth, hardwired at birth, say that you're going to be susceptible only to about 1,000 different stressors over your lifetime before you have some type of break, be it an immunological reaction or a reaction to an emotional stressor, and you become either extremely sick or you become, forgive me, let's say you turn into a classic schizophrenic. Okay. It's about your body's capacity to deal with cumulative stressors at that particular point, and whether it's Lyme's or some environmental insult other than a biological pathogen, it could be anything that might produce a similar trigger. So in other words, it's about susceptibility. Lyme's is just one pathogen. There could be Borrelia or a virus. And many individuals, when you look at medicine, and individuals who become sick after exposure to viruses, they oftentimes have debilitated immune systems as a result of that. They have fatigue, they have illness, they have cognitive difficulties. There's a huge constellation after that environmental insult, after that pathogenic insult of the virus. Now, was this some type of uber virus or super virus that this person? Not on average. On average, that viral pathogen was actually the trigger for what was already a debilitated and intolerant immune system. And that stemmed from the genetic blueprint that certainly could have been affected by methylation status. So following the dominoes, we don't want to say that it's Lyme's per se. There are a variety of different pathogens, viruses, bacteria, spirochetes, a variety of things that can cause huge oxidative stress, tremendous inflammation, huge debilitating fatigue on individuals after exposure that are really rooted upon interaction of all those factors and what was originally the basic biologic blueprint that has as its foundational markers deviant biochemical or deviant methylation markers. The short version would be a lot of stuff could really be affecting your body because of basic methylation in your blueprint. Okay.
So do you kill the pathogen and then deal with the methylation, or do you do both at the same time?
You do both at the same time.
Okay.
By the way, I want to make something clear. When the pathogen comes into place, it's not only the direct effect of the pathogen interacting with the methylation status. There's now the ramifications of that that pathogenic insult. It sets into play a series of inflammatory processes that are now outside of the realm of methylation. You kill the pathogen, you deal with the methylation, but still, if the inflammation is not dealt with, that patient may still experience tremendous debilitating effects. So you have to look at the incident of that pathogen in addition to the methylation and dealing with the pathogen itself. Very important.
Can the primary symptom of insomnia be classified as over or under methylated or pyrrole disorder?
I'm going to have to say no. And the reason why is because there's so many different reasons for insomnia out there that I don't think it's fair, forgive me, I don't think it's fair to insomnia to suggest that it can be lopped into a methylation disorder or not. There's so many different causes for insomnia. Certainly, looking at methylation status can be important, but there are a lot of environmental issues, a lot of, and forgive me, mental issues, stressors, worrying about your life circumstances, your job, interruptions in the sleep cycle, children, medical residency, a variety of things can lead to insomnia. Being a light sleeper. So it's not a single issue, it's a multifactorial phenomenon. Insomnia is one of the most common symptoms that is brought into any medical clinic, not just ours. And there are just so many people with this. Truly, the person who can fix this problem is just going to be a multi-trillionaire. But the answer lies not just in one area, it's really a multifactorial phenomenon.
If a person has been successfully treated for undermethylation with nutrients and supplements, will they still test for high histamine like they did prior to supplementing?
You will, in many cases, see, now, histamine is a reflective marker of methylation, so that I want to say. Don't try to follow numbers and don't be upset if the numbers don't change radically as your symptoms improve. The key is that your symptoms are improving with treatment when the original element showed you that you were undermethylated, or overmethylated for that matter. Okay, many individuals do show a normalization of those numbers with regard to histamine. There are some individuals who do not, but their symptoms improve. So keep that in mind when you're assessing the histamine protocol that you're dealing with. We know folks out there who are numbers chasers, and they become very obsessed with seeing numbers change. And we have to sort of emphasize that in this particular situation, they might be individuals where the histamine is more reflective marker than it is something to be followed over time. Okay, so.
Is bipolar disorder most cases?
Well, that's a very good question. And the reason I'm sort of pausing here is because, first of all, my question becomes, is that an accurate diagnosis? Unfortunately, we've seen way too many people who in the 1970s were diagnosed with schizophrenia. Then in the 1980s, those same schizophrenics were diagnosed as bipolar. Then in the 1990s, those people were diagnosed as bipolar with psychotic features. And then in the 2000s, we're back to schizophrenia and bipolar and a variety of other splits present. So I truly hesitate to sort of say bipolar per se, but if we must, what we would say is that usually there is indeed a biochemical contributor to that diagnosis. It can be pyrrole disorder, it can be high copper, it can be overmethylation, it can be undermethylation. It can be, now this we haven't talked about too much, it could literally be evidenced by a gut phenomenon that is caused by an extreme food sensitivity that has to be determined. Now that's only about 5%, so it's not the major group. But these are all very, very, very important elements. And so what I'll say is just in a short way, yes, it could be a methylation issue.
How does overall methylation status relate to global hypomethylation and specific hypermethylation in cancer context?
Ooh, wonderful. I love that question. Dana, I'm going to ask you to repeat that question for the audience and for me one more time. I want to get into this here. Well, what I'm going to say is let's talk about, now we've talked about really sort of the emotional mood disorders, cognitive states, and methylation. Let's talk about cancer for a minute. And ladies and gentlemen, I have to tell you, this is an area that is truly dear to my heart because I lost two good, close friends, colleagues from my medical school class long before their time to breast cancer. And it has so bothered me that when we started looking into the research regarding cancer, cancer treatment, and the methylation issues regarding cancer, it prompted a lot of research on our part and the development of supportive therapies for cancer individuals that are nutrient-based. So let's break this down for a minute. We know actually undermethylation phenomenon. Now, I want you to understand this piece. Methyl is protective of cancer gene expression. In other words, it's like looking at a blueprint of your entire genetic capacity, and then there's a machine that comes along and reads that blueprint, and everything that it sees, it puts into production. Okay? So we've got this machine that reads the blueprint that produces everything it sees in the blueprint. Methyl comes along and it covers up cancer genes. So it's like spreading a blanket over the cancer gene so that the machine doesn't read the blueprint showing cancer genes. So methyl is actually protective of cancer. Now, when we first heard about this, this was actually in the excuse me, Archives of Hematology and Oncology, Cancer Research. And it was actually Dr. Walsh that brought this to our attention. In his research, he saw that cancer therapies, or excuse me, cancer research was revealing that many of these individuals with undermethylation seem to have a higher incidence of different cancers. Now, those of you out there who know you're undermethylated or believe you're undermethylated, do not panic. This does not mean you're going to get cancer. But when we look at the research and the ideas behind it, what we're seeing is that methyl does indeed protect against cancer gene development. It doesn't mean it's 100%, but it's protective. Now, I want you to take those statements and I want you to put them aside for a minute because I want to talk to you about another concept. In the early research that was being done, the cancer patients who were done in this research piece were found to be undermethylated, and so the researchers decided, now let's go ahead then and try to treat the undermethylation and add more methyl groups. That might be a key to help cancer patients doing better. Well, what they found out was that when they provided their treatment protocol, these cancer patients did worse, and many of them died. Now, I want to tell you, the treatment protocol they used involved folic acid. Okay? We know from our work in autism and undermethylation in general, folic acid is what you do not use in order to treat undermethylation. It does not provide methyl groups. It actually gets rid of them. Now then, some of you scientists out there are saying, no, that's not right, that's not right. Folic acid works in different ways within different parts of the cell. Outside of the nucleus where your DNA is, folic acid is a wonderful donor of methyl groups. It provides methyl, yes it does. But in the nucleus where DNA sits, folic acid goes into the nucleus and it actually strips more methyl than it provides. So the net effect is methyl loss. Now remember, it's your DNA that's providing the blueprint for all your proteins, your enzymes, and your hormones to be produced. So if the markers, which are methyl, okay, are being removed in sum total by folic acid, folic acid is a functional demethylating agent at the level of the nucleus. Outside of the nucleus, in the area we call the cytoplasm of the cell, folic acid is a great donor of methyl. And this is the piece that most practitioners out there don't yet know or understand. It's folic acid having different capacities, different utilities, and different functions in different points of the cell itself, both the outside the nucleus and within the nucleus. The cancer researchers, back to them, were on the right track. Methylation was actually very, very important, and they were correct about the undermethylation. The problem was that they used the wrong agent to treat. They should have used a methylating agent like methionine or SAMe or something along those lines that would indeed give methyl. They would have protected these individuals, and they likely would have done better statistically. Now, let's say you folks are out there saying, you know, Dr. Mensah, I don't believe that, I don't agree with you, da-da-da. I want you to think about a drug that was used in cancer treatment that literally would deplete the system of folic acid. It was a wonderful drug called methotrexate. Any doctor will tell you that methotrexate was a great cancer agent, but it would strip the body of folic acid. So what did we do at the time back in the 70s and the 80s? We would give our cancer patients methotrexate and then add folic acid on top of it. And we're wondering why our patients didn't do better. Folks, I submit to you, in retrospect, the methotrexate was working because part of the mechanism was that it got rid of folic acid. We didn't realize that at the time, but now we see in this latest research the error of our ways, and hopefully we'll adjust those. Cancer in its forms has taught us some very interesting things. Now, we're not in cahoots with any of these other researchers in hematology and oncology and cancer research, but we're seeing the same stories being told about how folic acid interacts with methyl, how patients are responding to different therapies that involve folic acid and methylation, et cetera, et cetera. Folks, we can't all be wrong in all these different circles. The stories actually meld together and support each other, whether it's methylation issues in cancer, whether it's methylation issues in Alzheimer's, whether it's methylation issues in Parkinson's or autism. We're seeing the same effects with the same therapies, that is, improvement. So hopefully that giant long-winded story that was segmented in 3 different parts has kind of addressed the question and the issue. Methylation has a huge role, we're finding out, in oncology and cancer, and we're hopeful that now with some of the therapies that we're initiating will be even more significantly prominent as far as stopping this devastating disorder. There are supportive regimens right now that are being utilized that are nutrient-based, that are literally stopping cancers from growing, providing strength and support to the physiology of the person while they are battling cancer, and a whole lot of tremendous and wonderful outcomes. Many of the doctors that we share this information with abroad are coming back telling us that their cancer patients are not just survivors, they're thrivers. And so we're really pleased that all these areas have come together to provide us natural support mechanisms for individuals who've got cancer and have moved through chemotherapy or radiation therapy and are now doing significantly better. I do want to state that some of those therapies that are nutrient-based cannot be done concurrently with chemo, because many of them involve stopping cancer cells from growing, and as well as good cells from healing. So you kind of have to wait until, if you're undergoing radiation, for all that to be done, for the cells to start to heal themselves, and then those nutrient therapies can be applied. But everything is very case specific, but my long-winded story is that Yes, indeed, there is tremendous work that's being done in the field of cancer, whether it's breast cancer, colon cancer, or any other types of cancers. Many cancers are being addressed rather successfully with other supportive elements in addition to traditional modalities, and these other modalities are nutrient-based.
You speak a lot about methylation, but does this encompass all the cycles that span off from there? Do you supplement those as well?
Firstly, we target the methylation cycles, okay, and rather the end product of methylation. That's where, in many respects, it begins. Now, for individuals who, say, have an anxiety disorder, and we find that they've got metal dysregulation, they've got inflamed GI issues or gut issues, they may have methylation as well as pyrrole disorder, we treat all of them concurrently, okay? It is virtually impossible, nor is it really feasible, to try to follow every single body after you've dealt with methylation. See, the key point that we talk about here, I liken this to a domino effect. You know that if you start with the initiating domino and knock it down, it hits a series of dominoes that hit series of dominoes, and they hit series of dominoes. All you need to do is righten the first, or at least in the worst case scenario, second or third domino, and you actually stop the process from occurring thereafter. Okay? What we see in many of these conditions that we talk about with respect to methylation or anxiety or bipolar or schizophrenia is that there are consistently the same key elements that are the initial dominoes in several different systems. So systematically, you approach the prime movers, as we call them, righten them, and then they take care of the rest of the dominoes and other systems themselves. And that's what you see over time. So you don't have to chase the ongoing effect. Stop the initial outbreak, stop the initial flow, and everything else corrects itself.
Mm-hmm. Is there a difference between folic acid and folate from vegetables in healing methylation?
Functionally, no. Any form of folic acid, folate, methylated folate, unfortunately, well, fortunately and unfortunately, depending whether you're over or under methylated, they're all going to function similarly in terms of time. Folinic tends to cross the blood-brain barrier much more rapidly and more easily, but nonetheless, if you an overmethylator, a diet rich in folates, dark green leafy vegetables, soy protein, those fun sorts of things, that's going to be great for you. But be very careful if you're an undermethylator. That same diet can be extremely detrimental for you. Okay? Now, what I'm speaking about is not the average person out there, not the 70% of the population that is normally methylated. You folks can eat anything you want, okay? You do whatever you want to do. It's all about maybe your heart or diet or whatever you want to do. But for those who have cognitive disorders, that's the target audience that I'm referring to with this comment. If you have cognitive issues and you find you're either an under or an overmethylator, that's going to be the determining factor as to the types of dietary protocols you should follow. One size fits all does not work in these circumstances. Neither for your body necessarily translate into good cognitive health. See, we're all sort of told, and it's really true, vegetables are good for you, and good for colon health, and good for a variety of things. So lots of vegetables, lots of vegetables, lots of vegetables. That is great. There's nothing wrong with that. Okay, it helps clear the colon of toxic elements, removing debris, gets the bowels moving, all sorts of wonderful things, lots of great antioxidants. No one can deny or challenge that. However, if you are somebody with an absolute undermethylation issue and a cognitive disorder, anxiety, depression, schizophrenia, bipolar, etc., if you have a diet rich or just solely present with vegetables, dark green leafy vegetables, diets rich in folates, statistically, you will get worse. Okay? Nutritionists who are actually shocked by this, but they found it to be true themselves. And individuals who have come to us, who've talked about them with their cognitive health issues, moving to try to lead a more purified diet that they deemed was vegetable-based, they would often talk about how they got worse on those diets and didn't do as well. So if there is a cognitive issue in place, find out what your methylation status is first before you decide on which diet you should follow. Okay?
Mm-hmm. Do probiotics play any role in methylation?
Well, they are great in terms of gut health. In terms of being prime movers in correcting methylation disorders, not really. Okay. They are, once again, good for balancing the gut. Inflammation, great in general. It's good balance that's necessary for decreasing the inflammatory effects of a dysbiotic phenomenon. But overall, with methylation, that's not a prime tool that we use, not to treat methylation disorders.
Can you comment on Alzheimer's or MS? Do you work with any MS patients?
That's a great question. Right now, I don't want anyone to think that we are simply naysayers when it comes to genetic testing. We are not. I wanted the bulk of people out there, I wanted everyone to really sort of understand the real utility, the when you do and when you don't use genetic testing in terms of expecting a way to move about treatment or diagnosis. MS and Alzheimer's are two of when you do want to do genetic testing. There's a lot of great research out there right now that is actually pointing to different elements in the 23ME that are highly corroborative for MS potential and Alzheimer's. We're now looking at the role of genetic testing in Alzheimer's as far as predictability is concerned. Right now, because we don't have genetic treatments, it's not guiding treatment therapy. Our treatment therapy is geared in a different direction. But in terms of correlation, there seems to be some really strong evidence for genetic testing utility. Now, when it comes to methylation, what we do see, once again, is that with undermethylation, we tend to see in some family history different individuals with Alzheimer's disease. Not 100%, but it is a variable in Alzheimer's. With regard to MS, that's something that's currently being investigated, but we do not as yet have definitive protocols around genetic testing that are guiding us to treatment with regard to MS. Right now, I think in traditional medicine and in the orthomolecular realm, we are looking at as many anti-inflammatory and antioxidant therapies that come in several different ways, shapes, and forms as possible to deal with the issue of MS. I would not necessarily poo-poo any of them. Actually, I think it's a great thing looking at the different elements that are coming up for these treatments. The problem is that we don't have absolutely, a 70, 80, 90%, 100% cure or target rate for this. I think we're still all. It doesn't mean we don't continue to try as a medical community. It's just that right now we don't have a protocol around MS in order to boast of high levels of success in stopping or reversing this condition. Now, there are a few people out there who have mentioned that they're having tremendous success, but we really don't know yet of the statistical efficacy of those particular protocols. We're very hopeful. Individuals are using glutathione therapies. They're using a variety of things to deal with MS, and I'm hopeful. I don't care who does it, as long as we get that fellow licked and we can move on and help the individuals who are suffering with these disorders. But yes, indeed, we know that methylation does play a role in these elements. We just don't know, at least in the case of MS, to what degree that's guiding us. Okay.
Why, again, do you limit folate in undermethylation? And then...
Okay, hold that. Let me answer that part of the question. You limit folate because you're already low. Remember, let's talk about concentrations of methyl. That's what it's all about. That's what undermethylation and overmethylation are. Having too little methyl. Remember, methyl activates or inhibits. It starts or stops enzymes, hormones, neurotransmitters, genetic expression, neurotransmitter transport molecules can be either increased or decreased by way of methylation and methylation disorders. So methylation is really the stop-start button for a tremendous number of reactions and processes in the human construct. So methyl is important. If you don't have enough methyl, you can't have a fairly healthy working system, both in terms of body and mind. Okay? If you have too much, you can have difficulties as well in processing. So let's start with the idea that what we're talking about here is concentration of methyl. If you have too little methyl, you don't want to lose more of it. If you have too much methyl, you don't want any more of it. You want to get rid of it, okay? Or at least get rid of it enough to where it's at normal levels. Certain elements are going to increase methyl in the system. Certain elements are going to decrease methyl in the system. And we're talking about nutrient approaches. We know that folic acid, in its sum total, is going to decrease methyl in the nucleus where all of your directions for enzymes and hormones and neurotransmitters are basically created. So that's why you want to limit folic acid if you are an undermethylator in general, okay? But at the same time, if you've got tons of methyl and you're an overmethylator, you really want to give something or take something that is going to get rid of that methyl because you've got too much, and that's where folic acid has a positive and beneficial effect statistically. It's about concentrations, okay?
Do high fats with no or little folic acid play any role in methylation?
Do high fats with, can you repeat that question?
No, with little to no folic acid play any role in methylation?
Okay.
High-fat diets. I'm so sorry.
Oh.
High-fat diets.
Oh, I see. Do high-fat diets play a role in methylation? Okay. Not directly. High-fat diets tend to have effects in terms of oxidation and in terms of inflammation. Okay. And then now, of course, I know people are talking, we've got lots of books that talk about the necessity of fats, and if you're eating, a fairly balanced diet, usually you're getting your fair intake of fats in general. If you're pyroluric, you have to be careful. You can't really do tons of the omega-3s. You need more of the omega-6 fatty acids. So now, where is all this coming from, or going to, rather? When you're talking about fats and the diet and methylation, if you've got certain biochemical disorders, say pyrrole disorder, you have to be careful the type of fats that you're taking in, because that can worsen the pyrrole disorder, which can increase inflammation. Now, that increased inflammation can then make the entire process of methylation more challenging, because inflammation can affect genetic expression at the level of the DNA. Okay? So indirectly, high-fat diets or the wrong fats that are taken into the system, this is very individually based, those can have an indirect effect on the system that methylation is trying to work on. Okay?
Time, how quickly do you see the negative effects of a high folate diet in a person that is undermethylated?
Oh boy, let's talk about categories on that one. Okay, you can see this effect in less than 24 hours, depending on the individual. An individual who has autism, an individual with anxiety, depression, we've had many individuals comment that within a few hours after taking the wrong supplement, in certain cases, say folic acid or a certain type, they felt worse immediately. There were others who said, well, you know what, I initially felt better for a little while, and then I really began to plummet. And so when I stopped taking that particular folic acid supplement, I leveled off again and I did better. So it's different for different individuals, but it can be as quick as 24 hours. It can be as lengthy as within 2 to 3 months. Okay. But generally speaking, if it's not working for you, you're going to know it pretty quickly. Okay?
Mm-hmm. If someone with an MTHFR mutation has a high RBC folate level, along with low homocysteine level and low creatine level, is that indicative of a methylation issue?
I'm kind of, you guys can't see this, I'm kind of smiling here. I want us to repeat that question in pieces here, because my initial construct is to say yes, but I want to make sure I have all the pieces to that puzzle correct before I answer that question.
So they have an MTHFR mutation,
Mm-hmm.
high RBC folate level.
Okay, now let's pause. The MTHFR mutation, whether it's heterozygous or homozygous, in this case, may not be a player at all. Okay, it is not the sum total of the total of the struggle between overmethylation enzymes and undermethylation enzymes, giving us a net effect. It's simply one enzyme that may or may not have a qualitative effect, not a quantitative effect. So let's remove that one from the issue. The next piece to that, Dana, was what?
High RBC folate level,
Mm-hmm.
low homocysteine,
Okay.
and low creatine level.
Okay. The low creatine level may actually indicate an undermethylation syndrome and not an overmethylation syndrome. Creatine, remember, creatine is coming as a product of the sum total of all methylation. Okay? So if that person is low in creatine, we have to ask the question, why are they low in creatine? Remember, creatine is made from a biochemical consideration of all the enzymes of methylation and then that methyl moving through that process, as we showed you on a previous slide, to eventually come to creatine. So the one key piece here that's most informative is that they have low creatine. And as a result of that, I would suggest that this person is indeed undermethylated. So now let's go back to, this is going to sound kind of backwards, let's go back to that discussion about the MTHFR. The MTHFR enzyme may have played actually a more critical role in this case, but we don't know that without knowing what all the other enzymes are doing in total. It may be that it was not just MTHFR, it could have been the methylene synthase enzyme, it could have been the GMAT enzyme, the AGAT enzyme in the overmethylation processes. There were actually, so we could say that many of the enzymes in just the overmethylation pathway may not have been working at all, which means that you would therefore have low creatine levels as well, and in actuality, it's because of them not working that you have undermethylation. So really, all the enzymes in the undermethylation pathway could have been working properly, and none of the enzymes in the overmethylation pathway could have been working, and you end up with low creatine, and therefore you'd be undermethylated, primarily because your overmethylation enzymes weren't working. Now, folks, I know that may have sounded convoluted to many of you. But what I'm saying is that there could have been an enzyme defect, not just in the undermethylation pathway, but it could have been a defect in the overmethylation pathway that actually led to undermethylation and low creatine.
Can pyrroles and over or undermethylation occur simultaneously?
Absolutely. Pyrroles are not methylation constructs. Pyrroles are genetic elements and or environmentally induced, and they are separate and distinct from methylation. Okay. Treating pyrroles does not correct methylation. Treating methylation does not correct pyrroles. Okay. These are very different elements. So, indeed, they can occur simultaneously. They can occur one without the other, or you may have one, such as methylation challenge, and then develop a pyroluria later, given the right stressors. But they are not causative or relationally causative or even related constructs. They're separate.
Okay, this is our last question for the is overmethylated. I know they need folic acid. Should they stay away from methylfolate, the so-called better form of this vitamin?
Well, it's not that it's a better form. First of all, the answer is no, they don't need to stay away from it. It's not a better form. It's simply a different form. The idea was that with the methylated aspect added onto the folate, that you get a better absorption, quicker penetration of the brain, and ultimately a better effect. And that was unfortunately misguided in that they were actually trying to treat undermethylation when they started using elements like Deplin, the methylated folates. But in terms of an overmethylated individual, no, it's still a fine treatment protocol. There's a lot of good utility for Deplin in those cases, and it's simply another good piece, part of the armamentarium for dealing with overmethylation. Okay. But, once again, please, ladies and gentlemen, have a skilled professional deal with these issues for you. Do not try to treat yourself at home with these things. These elements are just a piece of the puzzle that is superimposed upon medical evaluation of your entire system. Okay? We're focusing on methylation today, but methylation is just part of the picture that sits within what we would consider to be not simply a patient, but an entire human being. It has its ramifications. So please be careful. Work with a skilled professional. Okay?
Thank you all for joining us. And if you're interested in becoming a patient of Dr. Albert Mensah, MD's, you can see us at our main clinic located just outside of Chicago. And for those who didn't see the slide earlier, this is our outreach schedule. Maryland in September, in California in November, and in Scottsdale, Arizona in December. And if you have additional questions for us and we weren't able to address them tonight, please email them to dana@mensahmedical.com. We'll try our best to answer your questions. And please, for more information in regards to research, additional videos, and other helpful information, please go to our website at mensahmedical.com. I think you'll find it very resourceful. And to really get an in-depth understanding, please purchase a copy of Nutrient Power. It can be purchased from our website, mensahmedical.com. 100% of the book proceeds go to Dr. Walsh's nonprofit charity organization. Thank you all and good night.